Hey guys, so today I'll be talking about biochemistry, enzyme kinetics. It's going to be great. I'm going to use a lot of extra videos. I'm just going to put them in here. I figured out a good way to download them and put them into videos, so I will be doing that. I will introduce them though as they come up. But first, we need to talk about enzymes. We're going to talk about enzyme kinetics. We need to talk about enzyme components. The major portion of an enzyme is called the apoenzyme. It's a super confusing set of nomenclature, and I apologize. It's just how it goes. The apoenzyme is the main protein piece that you're going to encounter. Okay, that's what we call the apoenzyme. It's the main protein portion, and then you have all these extra things. So cofactors are basically anything that we say is in addition to the apoenzyme. So these are things that are not protein, but they will basically make the catalytic site or the active site of the enzyme itself to be able to grab onto the substrate. So remember, we're making enzymes here, and they're going to have an active site, and that's the site where it does stuff. And so you have to have that active site properly formed. And so your active site or catalytic site needs to have not just the apoenzyme, but anything else. So coenzymes or cofactors, inorganic ions. I'm going to talk about a couple of those. But once those all come together to form a proper active site, that's called your holoenzyme, or some people say holoenzyme, but it's holoenzyme. And so that's where you're going to actually put your new molecule to be modified. Now, your enzyme cofactors, they run the whole gamut. We could spend entire class periods just talking about all the different cofactors that can exist. But it's basically the non-protein components that form that piece of the active site. Now, if they're permanently attached, we call those prosthetic groups. Okay, and if they're not permanently attached, so if they're temporary, we call those coenzymes. And there are lots of those, and some of those names are going to come up. So coenzyme A, I'm going to talk a little bit about. Biotin shows up a lot. FAD and FADH2, or there's NAD and NADH. We're going to talk about those. Those are awesome signaling molecules slash those are the molecules that we're going to use to take hydrogens and put them on the other side of the electron transport chain. But they also act as cofactors, which makes sense because you want NAD inside the enzyme that is going to then make NADH. And so it's thus a cofactor. But it's temporary because you want it to be able to come out of the enzyme. If it was going to stay stuck in there, then it'd be a prosthetic group. Lots of them also have vitamin precursors. So there's tons of vitamins. I think I have that on the next slide here. I used to have a better picture, but the quality was so low, and I could not find a good list of vitamin precursors. But anyway, just so you know, a lot of the B vitamins are precursors to many of the enzyme cofactors. Hence why you need a lot of vitamins in your diet. If you're eating a normal diet, like honestly, you don't need supplements. But if you're in some kind of place where you're just not getting the nutrition you need, then this is one of the problems. This is why we supplement things like cereals or pastas or things like that with vitamins and vitamin precursors, because they are the precursors to coenzymes. And so many of your enzymatic processes, especially in metabolism, so like the citric acid cycle and glycolysis, don't function if you don't have the precursors. We're going to see how inorganic ions, so I've got a bunch here. Notice that most of them are positively charged. They are going to function within our active site. Okay, so here's our active site. These cofactors are going to come in. And remember the function of these. You can think about this in a bigger way, like, oh, these are the things that come together to form the active site. But what we're going to get into is some more details of what's exactly happening. What is zinc's responsibility or magnesium's responsibility in this actual enzyme? Well, usually what it means is it's there to balance out charges, or to sort of allow the negative charges on like some oxygens. It can sort of pull that negative charge away and allow us to mess with some of those bonds. We're going to go through a few examples. Now, there are lots of, I put a few on here. There's tons and tons of cofactors. There's organic ones, there's inorganic ones. They have all kinds of things. Clearly, there's a lot going on. You do see some consistencies. So like, for instance, if you need to do electron transfers, you're going to use like flavins, which that's like B vitamins. If you're going to use do transaminations, then you need pyridoxal 5' phosphates, and these are the enzymes that use it. But there's lots of enzymes. If they have that group within one of their domains, then that's probably the job that it's doing. Now, it might be acting on a different substrate each time, but hey. So one example that I wanted to talk a little bit about is coenzyme A. We're going to encounter CoA all the time. So we'll encounter it as acetyl-CoA. And so here's coenzyme A here. So we can see it's got an adenine. So we got an adenine here with phosphate. So this should look very familiar. That's his ADP right here. Got an extra phosphate here though, so that kind of makes it look a little different, but we got an ADP here, okay, 2 phosphates. And then we're going to put a pantothenate, which it's just a really long carbon chain molecule. Now, technically this is not a cysteine. It's pantothenic acid, I believe is how you say it. But I like this diagram because it shows cysteine here, because I want to emphasize that there's a thiol group. So if you remember, a thiol group is when you have a sulfur on the end, and cysteine is one of the amino acids that has a sulfur, and it looks like this basically. And we're going to use that thiol group, and we're going to come over here. So when we actually make that's coenzyme A, but usually we're going to take pyruvate, and we're going to attach it to that sulfur here. Okay, we're going to form an acetyl thioester bond. We're going to lose a carbon. So pyruvate has 3 carbons. We're going to lose one of those to carbon dioxide, and then we're left with a 2-carbon acetyl group. And it's going to stay stuck here, and that is going to allow us to move this entire molecule, mostly we want this guy, into the mitochondria where we can do stuff to it. So we're going to take our pyruvate here, we're going to react it with the thiol group, so that's that sulfur on the end, and we're going to catalyze this reaction. We're going to move it using a massive complex of enzymes. That's going to actually do this and attach this here is a massive complex. There's 3 giant pieces. We're going to talk more about those when we actually get to this step in. It's between glycolysis and the citric acid cycle. And so we're going to go through that in much more detail. But the key here is that we are going to take our acetyl group, there's our acetyl group, stick it onto that sulfur, and there's our coenzyme A. And so that's CoA, and that's one of our coenzymes. So there's coenzyme A right there. So acetyl-CoA carboxylase is one of the 3 enzymes that we're going to be using in this complex to actually do this. And you can't have energy occur from the electron transport chain without this step. And so it's super duper important, as are many of these things. Now we're going to see CoA show up again when we talk about the citric acid cycle. So this is that step that I just talked about, where we take our acetyl-CoA, there's our CoA right there, that pink guy, there's our sulfur, there's our coenzyme A. So it just says CoA, and you're like, oh, it must be tiny. No, it's huge. It's this giant thing. But it lets us get through the membrane between the cytosol and into the mitochondria. Okay. But we're also going to see CoA show up again down here. So when we're actually doing our oxidative decarboxylation, we're going to go from alpha-ketoglutarate to succinyl-CoA. So we basically take this guy and attach it to the coenzyme A there. So there's that sulfur and there's that CoA there. And we're going to attach it there. We're going to use that to proceed through the rest of this. And so we're going to get some phosphorylation going on. It's going to be great. But I just wanted to show you that this is really, really important. So now let's talk about enzymes a little bit more. Specifically, we're going to talk about enzyme active structure. So let's go to a video.
Enzymes bind to their substrates, hold them in such that chemical bond breaking and bond forming processes take place more easily. Enzymes have a specific place in them called active site, where the substrate binds and real-time action takes place. Active site has a specific size, shape, and chemical behavior rendered to it by specific arrangement of amino acids. Thanks to these amino acids, an enzyme's active site is unique only to a particular substrate. In addition to the active site, many enzymes consist of a non-protein part called cofactor. Cofactors may be cations, positively charged metal ions, activators, bound temporarily to the active site to activate the enzyme, organic molecules, vitamins or vitamin products, coenzymes, that join enzyme substrate complex temporarily, prosthetic groups, permanently enzyme bound. Many enzymes only perform their catalytic role when associated a coenzyme. An inactivated enzyme, protein, along with its coenzyme, non-protein, makes up a system called holoenzyme. To add to the complexity, scientists termed this inactivated enzyme as apoenzyme. And therefore, the equation becomes holoenzyme equals apoenzyme plus coenzyme.
Oh, it's always nice to hear a British person talk. You just kind of assume that they're smarter than you, probably because they have a better educational system than we do. So that's talking about just the basics of the active site and how it works. Something I want to talk about, so that's called free med education too, to give them a little credit. We're going to watch more of that video in a second. So there are two models for substrate binding. So we're talking about active sites and enzymes and all this stuff. So there are two main models, the lock and key model. And that's where is number one. And that's where we have our substrate. So in blue here, you can see our substrate. That's going to bind into the active site and then be acted upon. Either usually we're cleaving bonds or making bonds, or we're doing all kinds of stuff to things, adding things, taking things away, whatever. Now, the induced fit hypothesis, so I say models, these are really hypotheses. The induced fit is that your enzyme sort of exists in this relaxed state, that's its normal state. And then when you have substrate binding, that's where it conforms to the substrate. So it kind of forms onto it. So it's not sitting ready to take it. It sort of forms around it. It induced. So the fit is induced around the substrate. It does its job. And then the actual change in the substrate shape changes the fit of the enzyme itself. And then you lose the substrate. So honestly, it's hard to prove either one because it's hard to take a snapshot of things over time because these things are so small. Induced fit is the more logical and the more likely hypothesis. That's basically how we see enzymes work, but we do see lock and key systems as well. So it's basically both, but for the most part, the induced fit hypothesis is the one that we're going to see more often. And so let's cut to another video.
Models of enzyme action. Lock and key hypothesis. This is the simplest model to represent how an enzyme works. The substrate simply fits into the active site to form a reaction intermediate. Just like the key fits in its specific lock, the shape isn't changed here. Rather, the structure of the substrate absolutely complements the structure of the enzyme, like puzzle pieces. Induced fit hypothesis. In this model, the enzyme, upon binding of substrate, changes shape. The matching between an enzyme's active site and the substrate isn't just like two puzzle pieces fitting together. Rather, the enzyme changes shape and binds to its substrate even more tightly. This fine-tuning of the enzyme to fit the substrate is called induced fit.
I wish I could do animations like that. So let's talk a little bit about active sites. So within active sites, that's kind of a generic term that we use. Generic's the wrong word. It's a broader term than what I actually want you to understand. So think about the active site as that entire region of a protein that's actually able to do something. And within there are all kinds of different things. So it's kind of like if I have to cut a piece of wood, I'm going to use a saw, right, to actually cut it, or think like a bandsaw or something. There's something that does the cutting, but I want my piece of wood to be held down, and so I need some spots to sort of hold it in place while I actually do the work with the saw. And so that's the difference here. So within my active site, so that's like the entire bench where I'm going to cut my piece of wood. The catalytic site is where I'm going to actually use the saw, where the saw is going to come down and cut the wood. But the binding sites are more like the clamps that I would use to hold the piece of wood there. And so that's how we see the active sites actually function. And so in this case, here's a picture of lysozyme, which is going to cut stuff, and it's acting on peptidoglycan. So that should sound familiar from microbiology. So peptidoglycan is that surface. It's a protein sugar cell wall structure for bacteria. And here we have lysozyme, it's acting upon it. And so it has a binding site, a nice big blue binding site here that's going to hold onto it. But the actual catalysis, or the catalytic site that's cutting it, is much smaller, and it's in this region here. And so but that's entirely contained within the active site, which is entirely contained within the actual protein structure. A lot of slides, and in this presentation, I want to try and give you guys some real examples, okay? So let's talk a little bit about this example. So in methanogens, so there's a bacteria that they use methane to get their carbons and their electrons, which is pretty atypical. We don't see a ton of those. We don't do that. Right, we use oxygen and we use organic molecules to get our carbons, but some of them can use methane. That's fine. It's not the greatest electron source, but hey, it's something. You don't have a lot of competition when you're using methane. And so they have a special enzyme called methane monooxygenase within, and it has an active site here. Okay, so we're going to use our methane, and we're going to use our methane. We're going to use within the active site of the enzyme. We're going to basically break apart methane and use it to get stuff out. Now, what I wanted to show you, I don't really care what's happening here, but I wanted to show you that active sites can vary. And the variation of active sites is dependent on the amino acid residues in the active site. Okay, now that sounds really logical, right? Yes, like if I change what amino acids are here, that's going to change the functionality of the active site. That's absolutely true. And so in this context, I'm going to cut to the authors. I put the picture up, but it's actually a GIF, and where it's showing the variations here. So we have two different active sites. And so what they did was they just changed some of the amino acids that are available, some of the amino acid residues that are basically poking into this active site. And so you can see if we change some of the amino acids that are here, which it doesn't look like they're changing here, it's actually something else in a different spot. But basically I just wanted to show you, if you change the amino acids in the active sites and you change how they're structured, then you can actually change your functionality of your active site. You can make it better, you can make it worse. Usually you make it worse. It's just kind of how things go. Let's do another example here. So triose phosphate isomerase is a mouthful, but it's one of the enzymes that we're going to have to know in glycolysis, okay? And what it does is it's going to move this oxygen over here. You're going to basically see this hydroxyl group here, and we've got this double bonded oxygen here. We're going to flip them, okay? So it's an isomerization reaction, right? So I've just changed them. Nothing has changed except the position of these. So it's still the exact same chemical makeup. And it's going to go from DHP to G3P, okay? Which when we get into glycolysis, we'll talk more about that. Now, what's really interesting is the enzyme that does this, okay, which is triose isomerase, or triose phosphate isomerase, if you want. When it binds, so here, this is the active site, okay? So we got our active site here, and we have this nice red portion of the active site. And this is kind of showing more the induced fit hypothesis. So when our substrate comes in, so this is the dihydroxyacetone phosphate that is going to come in here, which is the longest name ever for like this little molecule. And it's going to come in here, and then when it actually binds into the active site, this giant portion of the active site is going to bend over. Okay? So it causes an entire strand shift of this amino acid to kind of bend in. And what that does is it stabilizes the intermediate forms, which we're going to talk more about what that really means. But what's happening is it's moving this big chunk of this protein into the active site, and that protects the DHP from being resolubilized. So it's soluble in water and it's floating around, and then the enzyme is sort of grabbing onto it, and it needs to hold it still. And in order to prevent it from floating away, basically, it sort of kind of clamps it down with this portion. And so its active site actually changes, which is really, really interesting. Let's talk a little bit about something that's pretty relevant. So I'm an immunologist, and so I deal with antibodies a lot. I worked on a vaccine. We were trying to make antibodies against HIV. And so antibodies themselves are a great way to look at active sites. They're not necessarily enzymes, but the same idea applies. They have an active site. And so in the case of an antibody, so here's your, you've got your heavy chain down here, and you've got your light chain, and you got your variable region here, and that's the business end of an antibody. So these two pieces right here look exactly the same, and they're going to grab onto an antigen. So that's basically any chunk of something. So antigens are really big, and then they have a portion that can be bound by the antibody. Okay, and so that's the active site. And so what we see is you can vary the active site structure. Okay, so this variable region, so the V stands for variable here, so variable heavy chain, variable light chain, that region we can change, and it's just what amino acids are there. And so if I change the amino acids, if I make some, they might bind better to an antigen, they might bind worse, that's bad. When they bind better, we actually tell the B cell that made that antibody, hey, you had really good binding, you should keep making more antibodies. That's really good. So there's like a positive feedback mechanism there, which is really interesting. Now I found another research group. It's the same research group that's doing this kind of work. And what they did was they were looking at antibodies, and antibodies, they're just proteins. Right? They're just really complex protein structures. They have quaternary structure. And you can change the active site. Normally antibodies only grab onto one thing. That's really, really important. Sometimes you can get broadly neutralizing antibodies. So that's like an antibody that could grab onto every strain of the flu. that would be really cool. Then we wouldn't need the same flu vaccine, or I mean, a different flu vaccine every year. If you could just get one, they could bind random stuff. And so designing antibodies and the actual structure of their active site is really important. And so in this case, they have the SPE7 antibody, which it doesn't really matter what it binds to, but most antibodies only bind one epitope. So that's like an antigen, like a piece of an antigen. But this one happens to be able to bind two. So there's like a deeper groove portion here is one form of it, and it's in equilibrium with this second form. So these pieces here, so you can see that we got some a beta sheet here. And so these beta sheets can shift and sort of dissolve, and then that makes this groove of the active site deeper, which allows it to bind one type of antigen, one epitope of an antigen, and then you can shift and reform those beta sheets, and you have a more shallow groove, which it doesn't look like a groove, but it's actually there. And it's a more shallow groove, which lets you bind a different antigen, which is really, really cool. So there's multiple bindings of an antibody. That's super cool if you're a huge nerd like me. Start moving on. We need to talk about what usually comes up when you're talking about enzymes, and that's enzyme kinetics. So that's your activity or your reaction rate or the velocity. It all depends on all kinds of things. We're going to talk generally in optimized conditions, but you do need to understand the idea that changing the actual conditions that your enzymes operate under can change how they function. And so that's why in biological systems, we tend to want to maintain temperature, right? We've talked about maintaining pH. The actual substrate concentration, we're going to focus on that a lot more later. But these are just really important ideas. So why are you 98.6 degrees in most of your body? Well, which is actually up for debate, but well, because most of your enzymes have an optimum temperature. So the optimum temperature is usually distinguished by you need the right amount of energy in a molecule to get it to vibrate sort of into the active site properly. But if it gets too high, then you're going to start breaking the hydrogen bonds, the disulfide bonds, the covalent bonds that are holding the structure of the enzyme together. And so up until your optimum temperature, like you're just basically vibrating your molecules until they like to get into the active site, they can be acted upon. But if you get too hot, I mean, you die. So you get denaturation, right? So the rate is going to go down if you're dead. Fun fact. But that also depends. So some enzymes prefer really hot temperatures. Some enzymes like cold temperatures. Usually that depends on the creature. So there are bacteria that live super deep in the ocean by these ocean vents where it's super pressurized and it's super duper hot. And so their enzymes are resistant to denaturing. And so they have a higher optimum temperature. Whereas us, we generally 37-ish degrees is most of our enzyme activity is happy at 37 degrees, though it depends. I mean, your skin is cooler. Your mouth tends to be slightly cooler, and so they have different optimum temperatures. The rate tends to go up. I just grabbed a few of these diagrams here. So you can see rate goes up. This is a percent of active enzyme. It drops because of denaturation, of course. And then this is basically the same thing. So as temperature goes up, this is why the activity goes up, because you have, your temperature going up, things are bouncing into the active site, but then denaturation. And it's usually faster is why I had this. It's not as obvious on this diagram that this is kind of slower and then this is pretty fast, but you have denaturation pretty quickly. But I mean, you have way bigger problems if you're stuck at this temperature for too long. Now, the other thing is pH. So we always need to talk about pH. It's going to come up in the big example that I'm going to go through later on. And your enzyme kinetics are super dependent, but it depends on where you are. And remember, pH and pKa, which is something we talked about a lot in that previous lecture, are really, really important. Enzymes are going to be made of amino acids, and amino acids themselves, their R groups, have specific pKa's. So either they're deprotonated or protonated, and that can have a strong impact on the active site. We're going to see that a lot of active sites actually contain charged residues, or either positively or negatively charged residues, which is going to depend on their pKa. And so you can see here, it depends on what your enzyme is. If you're a salivary amylase, you like being at a pH of pretty neutral, okay? But if you're pepsin, which is in the stomach, you actually function best at a really low pH. And so I want to cut to another video that talks a little bit about this.
Environmental effects on enzyme function. Active sites are very sensitive. They sense even the slightest change in the environment and respond accordingly. Some of the factors that affect the active site and consequently enzyme function include temperature, The suitable temperature for enzymes to function properly is 37 degrees Celsius. Increasing or decreasing the temperature above 37 degrees Celsius affects chemical bonds in the active site, making them less suited to bind substrates. Higher temperatures denature enzymes. pH. Amino acids present in the active site are acidic or basic. Fluctuation in pH can affect these amino acids, making it hard for substrates to bind. Extreme pH values can denature enzymes.
So I just like having a few visuals in there. So we need to talk about enzyme activity, and we need to talk about what enzymes actually do before we get too deep into that. So you may have heard, everybody likes to say this out loud, but they don't necessarily entirely understand what that means. Enzymes lower the activation energy. A more simplified view of enzymes is that they just make reactions happen. Okay, well, we need to talk about that. What does that really mean? Well, they lower the activation energy. So from a mathematical perspective, i.e. we're graphing it here and the amount of energetics that are actually there, what that really means is that it stabilizes the intermediates. So to go from a set of reactants, which could be one reactant going to multiple products, or multiple reactants going to a single product, or whatever, then you are going to have an intermediate in between those. And when you have intermediates, if you stabilize those intermediates, that makes it a lot easier to actually get through the reaction. Now, you could have a negative delta G, which is going to be favorable. You could have a positive delta G, which is unfavorable. But you can overcome that by combining your delta Gs, which we talked about this with the kinetics. We talked about kinetics before, with thermodynamics, I'm sorry, where we can string together really strong negative delta G reactions, like the phosphorylation or the dephosphorylation of ATP into ADP, with not as favorable reactions. And so we can just kind of string things together. ATP is going to be our main molecule to actually do that. And so that's going to allow us to either kind of push ourselves from our reactants into the products, or pull ourselves from the reactants into the products. And so if I keep pulling the products away, then that favors pushing them this way. If I keep building up the reactants, it can push it this way. And so, but enzymes are going to basically make it easier to overcome that activation energy. Okay, and so we need to talk a little bit about that. And so I wanted to show this next video. It's just a nice little visual of what's happening.
In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur.
That video because it's just visualizing what an enzyme is actually doing. Yes, it lowers the activation energy by stabilizing the intermediate, but what's it physically doing? Well, it's just making a nice, comfortable little active site to let things happen. It can have the spots to actually do the reaction as well, but it's like a nice little cradle spot, like a little nest to have things happen in. We're going to come back to that video. Shout out to the Protein Data Bank. It's a massive database of proteins and structures and things like that, and they made that video. And that's why we're going to watch another one of their videos much later, and they're going to show very accurate models because they're the protein database, and so they have really, really nice model systems. So we need to talk about enzyme kinetics. And so here's what I want you to do. I want you to think about this in a purely hypothetical way. So you have one enzyme molecule, just one. Okay, so we're going to have one enzyme molecule. And we want to see how many substrates, right? Because they grab onto a substrate. So imagine, I like to think about it cleaving something. So your enzyme is going to grab onto a molecule and cut it in half. Okay, awesome. So let's say that it takes, we're going to give it 1 second, and we're going to see how many it can cleave. Now we call this our velocity. In this context, we're talking about initial velocity because hypothetically, if you just kind of let your reaction go, everything would eventually slow to zero because you would use up all your substrate. So we're going to look at it right away. So we're going to say, okay, if I took 10 substrates and I gave them to my one enzyme, how many can it cleave in 1 second? Well, you know what? In 1 second, all of them were cleaved. Okay, awesome. Pretty straightforward, so it's cleaving pretty fast. Okay, so let's throw 100 of them at it. Oh, and it cut all 100. Okay, cool. Let's throw 250 of them at it. Oh, it almost finished. So in 1 second, it cleaved most of them, but it left 50 behind. Okay, let's try 500 of them. oh, hey, look at this. So it cleaved 250. Now that was more, so you'd think it was hitting its limit, but it hasn't quite reached its limit. It's able to fit a few more of those in there because I've increased the concentration that's going to kind of force more of them into that active site, but it's definitely slowing down. So let's try 1,000 of them. So I try 1,000, and it's actually a little higher here. We got 400. Okay, let's try 1,500. So we're really amping it up. Ooh, we got to 450. All right, let's try 2,000 substrates and see how many we get. 455. So man, I must be getting kind of saturated. Well, if we plot these kinds of velocities on a graph, we're going to see something that looks like this. This is a Michaelis-Menten graph. Okay? This is what it typically looks like. So on the left side, so on our y-axis is the rate. So this is often referred to as velocity. Okay? And down here we have our substrate concentration. So that's referring to 100, 250, 500, or whatever. So that's down here. And then how fast this is going is over here. And so you can see early on it's pretty fast. Okay? We start cleaving stuff really fast. As our substrate concentration increases, the rate of the reaction, it starts out real fast. So I try and use like real English when I talk instead of using like science jargon. So it starts going really fast. See that this angle is pretty steep, right? It's going really fast. The enzyme is ready for more. You can just keep giving it substrate and it's going to keep cranking them out. It's going fast, cruising. But once we start reaching kind of a mid-level here, okay, we can't add any more substrate. That doesn't speed it up because the actual number of active sites or the ability to of one enzyme to fit them into its active site fast enough, it's hitting some kind of limit. Okay, so I just can't fit any more in there. The enzyme can only do its job so fast, right? There's some kind of limit. And so we're approaching that limit, and no matter how much substrate I add in, I can only do so many. And so what we approach is called Vmax. So Vmax is the maximum velocity at which an enzyme, which is just a machine, can actually do its job. Okay, and so you could, it's kind of like, well, I have another analogy in a second. So let's talk just some of these. We're going to see this a lot, so get these things kind of in your head. So Vmax is the maximum velocity, okay? And then what we can do is we take Vmax divided by 2. So I divide my Vmaximum by 2. And that gives me the substrate concentration, right? So substrate concentration is down here. Right? How much substrate is here is down here. And so if I take my substrate concentration and I look up here at Vmax divided by 2, right? So there's Vmax, I divide that by 2, and I climb over here and I zoom down. Km is my substrate concentration, okay? And this is a constant for this enzyme. As long as there's not the presence of some kind of inhibitor or something, because we're not talking about that right now, we're not talking about inhibitors, we're just talking about enzymes, this Km is a constant. Okay? This is called the Michaelis constant, and it's a measure of infinity. So the Km for an enzyme will not change. Caveat, without an inhibitor. Okay? Now, it's all about binding affinity. So how well is my enzyme actually grabbing onto stuff? Okay? So if it's able to grab onto stuff super well and perform its job really well, then it's going to have a really small Km, i.e. this is going to kind of slide this way because it's able to grab onto stuff, do its thing real fast. Okay, so the rate is really fast, and so my Km is small. But if it takes me a while to grab onto each substrate, then my Km is going to be a little bit bigger. Okay? It's all about affinity. Because it's hard for me, it's just like this has always been challenging stuff for me, so I'm going to really try and break it down so that it's pretty straightforward. So if you have a really big Km, okay, right? So Km is the substrate concentration at your Vmax, half of your Vmax, which sounds like crazy science, not even science, it's like calculus jargon. It's how well you bind. So if you have a really big Km, so like for the red here, so we have a Km of like 1.5. So that means I don't bind my substrate super well. And so I can saturate that active site really fast, okay? So it takes longer to get the job done. I'm not grabbing onto my substrate really well. Now, if I have a small Km, so look at this rate. So that angle is super sharp, right? So I'm able to grab stuff and get my job done real fast. And so I bind my substrate, therefore, really well. So if I have a really short Km, that means I'm binding my substrate really well. I can do my job really fast. So for this graph, the red enzyme, and this actually is a real enzyme, this is for hexokinase, which we're going to encounter at some point later. So the red enzyme here, doesn't grab onto its substrate, so let's say glucose, as well as the orange enzyme does. Okay? There's different versions of hexokinase in your liver. There's cellular hexokinase and there's liver hexokinase. They have different functionalities for different reasons. So this red one, it doesn't grab onto glucose as well, okay? But it could gobble up more at the same time. So let's say there are advantages. Sometimes you don't want to, so in the case of glucose, sorry, I'm tripping over my words. In the case of glucose, sometimes you don't want to cleave the glucose as fast as other times. So if you're inside of a cell, yeah, I want to get that glucose chopped up and doing its thing real fast. However, if I'm in like the liver, maybe I don't want to chop it up as fast because I'm going to convert the energy from that glucose into like glucagon or fatty acids or something like that. So that's why there's different versions. So I don't want you to think that just because an enzyme has a low Km, so it doesn't grab onto its substrate as well, that's a bad thing, okay? Because that's a really common problem. I say common problem because that was my problem. So your problems become my problems. So in this case, the red one doesn't grab as tight, and the orange one grabs even tighter. However, notice they both have the same Vmax. If I increase the actual amount of substrate that's there, eventually they all get saturated and get pushed through at the same rate. Now, here's another analogy that I have. So this is the one I like to talk about. So if you have, let's say it takes you 1 second. If you have a hold of piece of paper in your left hand and you cut it with your right hand, so that's what's happening here. It takes you 1 second to do that. Okay, so it always takes you 1 second. Doesn't matter. Now let's say let's say we have 3 different people. So we have person A, person B, person C. So person A, using their left hand, grabs it really fast. It takes them 1 second to grab a piece of paper. It takes them 1 second to cut it, and then it takes them 1 second to drop it. So it's really fast. So in total, it takes them just like 3 seconds to do the whole process. Okay, awesome. So they have kind of like a medium Km. They grab onto it, they cut it, and they release it really fast. So this is just another way for me to think about Km. Person B is kind of slow. It takes them forever to grab the piece of paper. They cut it at the same rate, right? So just squeezing it to cut the actual piece of paper, squeezing those scissors, takes 1 second. Okay, fine. And it takes them forever to drop it. So maybe it's like some old lady or something. So they would have a really low Km. So it takes them a while to grab onto it and actually release it. So that's where that timing is coming into play. So they're just not as good at holding on to the paper. Whereas somebody, maybe this person's wearing like really sticky gloves, so it lets them grab onto the paper really well, and then they can throw it really hard and release it really fast. And so they have a really high Km. They're able to grab onto it really, really well. A weird analogy, but I don't know, that's one that kind of works for me. It has more to do with time. Okay, I want to cut to another example that hopefully will help. This is one that helps me. This is a master's project from a person who was getting a degree in like biological education, or no, it was biological communication. I think her name is Ursula. And she is going to use robots and how robots are actually functioning to convert forks into sporks. Hopefully you know what a spork is. I like this a lot, so let's watch it.
Let's pretend these robots are enzymes, tasked with turning plastic forks into sporks. A fork goes into the active site of a robot and is converted into a spork. As more forks are introduced into the system, robots convert these to sporks at an increasingly faster rate. However, there are only so many robots, and eventually the maximum number of robots will be active, and adding more forks won't change the number of sporks created. This state is called Vmax, the maximum rate of catalytic reactions. The substrate concentration, where the reaction rate is half of Vmax, is called the Michaelis-Menten constant, Km. Km describes an enzyme's affinity for its substrate, where lower values mean higher enzyme affinities for substrates. For example, our Robros in the top row have a lower Km, and it takes very little substrate to saturate the system. Our less efficient Robros on the bottom have a higher Km, so a higher concentration of forks is required before they're all active.
Something I wanted to show you guys, I'm going to have the link in the actual PowerPoint here, but I'm going to cut to it over here. Hang on. This is a Michaelis-Menten graph, and you can go here and mess around with stuff. So you go down here, you can't really see it. It says add to plot and then clear the plot. So this is our plot here. and just to fit it in, I had to do it like that. And so we're going to say, let's do 5. So, because I think one of the reactions up here was like 5. So our Vmax for one of our enzymes is 5. Okay, and so we're going to say, all right, our Michaelis-Menten constant, well, let's just do, so if I jump back here, so this 5 was our Vmax for this hexokinase, and one of them was 0.25 and one was 1.5. So let's jump back over here. So 0.25, 0.25. Add that to the plot. Cool. Okay, so it tells us our Vmax was 0.5, right? Or 0.25. That's our Km is 0.25. I'm sorry. And there's our Vmax, which is 5, and so 2.5 is our half our Vmax, so 0.5 Vmax. So that it tells you that there. And now we wanted to change our Michaelis-Menten constant, and so that, remember, that is a substitute for how well we actually grab onto it. So if I grab onto my substrate really, really well, I'm going to have a lower Kmax, or a Km. I'm so sorry, a lower Km. If I have a really big, if I don't bind it as well, I'm going to have a larger Km. So that other one was like 1.5, I think. Right? Let me check. Yeah, 1.5. So I'm gonna, but the Vmax is the same, so let's add it to the plot. Boom, there we go. And so our half our Vmax there, it's the same. It's cruising way along there, and you can click on here to kind of extend it out a little bit. That's a little easier. There we go. So you can see there's our Km. It's way out here. So our Km of 1.5, which the scale is just wonky. But our Vmax is the same. So eventually, if I change our scale here, eventually they all kind of approach that Vmax up here, but this one needs a lot. So it doesn't bind it as well. Okay? So that's what they look like. And I wanted to show this to you. You can play around with it, but also we're going to come back to this. So I'm going to clear the plot and we'll come back to this. Okay? So let's get back into the PowerPoint here. So I apologize for the quality of this. This is the picture, the diagram I like. It's from Khan Academy. And so I wanted to put this on here. So another way to look at this are kind of 3 different ways to look at this rate of reaction. And so as your rate of reaction is going on, you have plenty of active sites for your substrate. So you've got plenty of active sites for your enzyme to grab onto the substrate and cleave it. Okay, so that's kind of the first region. So this is more of a practical approach. Now, as you start bending this curve a little bit, that as I'm approaching my Vmax, then I'm having more competition, right? I can only grab onto as many a certain amount of substrates, and so I start to lose some of that. I can only do so much, and then eventually, most of my substrate or my active sites are full of substrate, and so I reach a limit, right? I know I keep reiterating all of this stuff, but I just some descriptions help other people, some descriptions don't help them, and so I try and give you lots of variety here. You might encounter too, there are other ways to plot enzyme kinetics. Most often you're going to see Michaelis-Menten plots like this. You can see Lineweaver-Burke plots. I've seen those before. Basically, they're kind of an old school way of doing this. So before we had computers that could basically do this kind of thing and do all the calculations for us, it was easier to, you would plot the inverse of the reaction rate versus the substrate concentration. So they're all like inverses here. And then when you had your intercepts, those intercepts told you things. So the intercept with the y-axis was 1 divided by your Vmax, so you could figure out your Vmax. The actual slope was Km divided by Vmax. And then where the intercept crossed the x-axis was your Km. And so it let you calculate those things. But the problem was that it was really prone to errors. And so now we just use computers to do it. And so there's different ways to look at it. And so you can imagine it's easier to look at this if you don't have a computer. If you have a computer that can just tell you everything, then you just do it this way. But I put these on there because, I don't know, you got to look at them. So let's keep moving. We need to talk about association, okay? So bringing things together, and dissociation, pulling things apart. Okay? So you think about, we have the active site of an enzyme. It's going to associate itself with a substrate. And so we get an enzyme substrate stuck together thing. And then we're going to dissociate. Okay? So some enzymes every enzyme that's going to grab onto the substrate, and there's a rate, a specific constant. We call that the association constant. And basically that's how well does it grab onto it. Okay? And then the dissociation constant is how well does it fall off? Or another way to think about it is how often does this, if this was an equilibrium, how often do we associate? And if this was an equilibrium, how often do they fall apart and dissociate? Okay? So there's different ways to approach it. Okay? If you have a really big Ka, so that's your association constant, that means these things like to stick together. So if this is huge, okay, which I'm going to demonstrate that visually in a second. So, but if your Ka is really, really big, then your Kd is small. They're inversely proportional, okay? Hence this equation here. Ka versus Kd, it's inverse. So if I have a really big Ka, my Kd is really small. Okay? So if this is 10 to the 8th, then this would be 10 to the minus 8th or whatever. And then if I'm, but the opposite could also be true. So let's say that my enzyme and substrate don't like to be together, okay? So they would therefore have a really small association constant. They prefer to be apart, but the Kd would be really big because it's favored to go this direction, okay? So let's talk about that. So if I have a really big Ka, say I made it really big, cool. That means more often I like to move to over here. So it's favorable, the constant to actually have my enzyme and substrate stuck together is favorable. So I have that happen more often. Whereas the opposite is also true. So if I have my Kd being really big, then my Ka is small. See, it's smaller there. And then I have lots of them, they're separated, right? They're not stuck together with them. See that nice little blending there? That's pretty cool. Fancy pants. And then I have these all separated. So they're just kind of floating around like that. So you can kind of bounce back and forth between these two, and that'll make you really dizzy, okay? What does it really mean? Well, all we're doing is to actually calculate the dissociation or the association constant, because they're just inverses of each other. So let's look at dissociation. So I say, okay, well, what's my concentration of enzyme times my concentration of my substrate divided by them together, the concentration of them stuck together. Okay? And then if I flip them, that's my association constant. And so how often are they over here divided by how often they're over here? And that tells you your association and dissociation constants. Now, this is not just for enzymes or for substrates. We can also do that for antibodies, like I talked about earlier. That's why I kind of mentioned that antibodies aren't enzymes, but they do have active sites that grab onto stuff. Just some background, B cells make antibodies. That's what this is for. I put a lot of extra stuff in here so that you can read it later on and not have to listen to me. But if a new site binds a pathogen better, okay, so if you have a really strong Ka, so we have the Ka and the Kds for these active sites, this variable region. This is where you want to grab onto stuff. So like if you get coronavirus or whatever, and you make antibodies against the spike protein, well, there's the spike protein, and then a portion of it can bind to the active site, or, I'm sorry, not the active site, but the binding site of the antibodies here. If it associates really, really tight, which is what you want with an antibody, so that means it grabs it really well. So that means more often than not, when you have antibodies in the presence of this antigen, they grab onto it, then you'd have a really big Ka. And the Kd would thus be really, really small. And when you have really big Kas for your antibodies and small Kds, that stimulates the B cells to make more of their antibody because it's working really well. And so they get a nice positive feedback. If it's the opposite, if your Ka for some reason gets smaller and thus your Kd gets bigger, that means you're not grabbing as tightly. Okay? So you're not doing as good a job, and so that B cell does not get that nice positive feedback mechanism, and so you wouldn't keep on making these kinds of antibodies. Hopefully that is helpful for Ka and Kd. It's something we always talk about with enzyme kinetics. Now, we need to talk about some fun stuff, and then we're going to get into some very specific examples, and then we'll be done. So we need to talk about inhibitors, which is always fun. So at the top here, I have what our normal reaction is. Thank you, Khan Academy. I didn't put the little thing here. I'll write it down. Khan Academy. So those are from Khan Academy. Thank you very much. They make good pictures. And so up here we have our normal reaction. There's our enzyme, grabs the substrate, does its thing. Okay, so in this case, it's cutting it in half. Okay, cool. Now, we have different kinds of inhibitors. Your body is going to use a variety of inhibitors. We can add inhibitors to you. Some toxins and poisons are inhibitors, so let's talk about this. So there are honestly two main types of inhibitors that usually get talked about, okay? Competitive and non-competitive. Competitive just means that your inhibitor is going to stick in the active site, okay? So the enzyme itself isn't affected, okay? So your Vmax will always be the same, so it doesn't really matter. But basically you have a competitive inhibitor. It looks just like, see this piece here fits in the active site, but it's blocking how much substrate gets in there. And so your Km is actually higher because you're sort of artificially filling your active site with this purple dude. So you want to be grabbing green ones. And so let's say I threw these in, they're blocking half the time you're accidentally grabbing a competitive inhibitor. And so therefore, you're just not grabbing as tightly onto these green ones. When you grab one, you grab it tight, but most of the time you're just not grabbing it well. And so your Km goes up, okay? And I'm going to show you what that physically looks like on a graph in a second. Now, when we have our different version, so a non-competitive inhibitor, which if you took micro with me, that would be like an allosteric inhibitor. I'm going to talk about those in a little bit because their kinetics look a little wonky. We also call those non-competitive, right? Because they're not competing with the substrate binding site. They're just competing somewhere else. And basically what they're going to do is they're going to physically change the shape of the active site. And so they grab on, and in this case it was bound to the substrate, and then this is the non-competitive inhibitor binds somewhere else on the enzyme, okay, and makes it so that you can't proceed, basically, or not as well, so you can't do it as good a job. And so you may be able to actually bind the same, you're able to bind the substrate at the same rate, so your Km is the same, but your Vmax drops, so you just aren't able to do as many over time, okay? And so, which might seem a little confusing, let me show you some pictures in a second. Uncompetitive is one that does not get talked about that much because it gets kind of complicated, but basically it's kind of both. And so I made this one, so I stole like this picture and stuck this on there, so that's why it looks a little different. Look at that, it looks almost the same. I actually changed, I added that little U there, pretty fancy, right? So an uncompetitive inhibitor is an inhibitor that likes to prefer to, it binds to the enzyme, okay, blue, enzyme, substrate, green, complex. So when it actually gets stuck in there, they usually are depicted as kind of grabbing onto here and sort of holding the substrate in there, okay? So they prefer when the substrate actually falls into the active site, that's when the inhibitor comes along and sort of keeps it in place. And so it's doing two things. It's kind of forcing the substrate to stay stuck, okay? And so that's going to affect your Km, just like the competitive inhibitor drops or raises the Km, which is bad. That means it's not binding as many. And then your Vmax will also go down because it's messing with the enzyme's ability to actually bind things. And so over time, its Vmax goes down. And so let's look at what these physically look like. Okay, these are just the same descriptions here. I just changed them. And so over here we see, okay, here is our normal enzyme right there in purple. Okay. So there's our normal enzyme. And then when I add a competitive inhibitor, right, so that's this guy here. So I'm going to compete. So my Vmax stays the same, right? So eventually I hit that Vmax. Right? However, my Km is going to be pushed over here, right? Because half my Vmax, so where's half my Vmax? So it's about here. Okay, so there's my Km would be right here. And so by increasing, having the competition there, my Km, so I'm competing, I'm not able to bind things as well because there's something competing with that binding. And so my Km actually is increased, okay, which in this context is a bad thing, though I also want to dispel the notion that this is like bad. I mean, sometimes you want there to be a competitive inhibitor. But usually we think about it in context of like a poison or something that you ate. Now, non-competitive inhibitors, right? So that's where they're like allosterically binding somewhere else and affecting the enzyme. Well, your Vmax drops, okay? It hasn't affected the actual binding, right? So your Km stays the same. However, because so let's see, half our Vmax would be like here. So there's our Km. Half our Vmax would be here, and they actually line up, okay? But the problem is our Vmax is not as high, so we're not able to do as much stuff. Okay, so what I'd like to do is jump back into the little diagram that we had, or that little sample one. So let's jump to that. We're going to kind of build our own visualization of what this is going to look like. So we're going to have 5 be our Vmax, okay, so we're going to kind of maintain what we've done. And then our Km, which is our binding affinity, is going to be 1.5. So let's put that on our plot. Okay, so there's our just regular, so blue is going to be our just regular one, and then we're going to do our competitive inhibitor. So when we add a competitive inhibitor, our Vmax stays the same, okay, but we are competing, so our Km is going to increase. So let's make it 3. Okay, we're going to double our Km there. So let's put that on here. So in red, now we have our competitive inhibitor, right? So if I extend this out, which is probably going to mess this whole thing up, but if I extend it out, our Vmax is the same, but our, it's all like weird now. That's fine. There we go. Okay, So we're going to eventually hit the same Vmax. However, our Km is extended out, right? So because it's just, we're having a competitor in here for that substrate binding. And so, or for the active site that binds the substrate. And so we just don't bind it as well because there's something else in the way. All right, so now let's try a non-competitive inhibitor. So that would be like an allosteric inhibitor. Now our Vmax is going to drop down, okay? So I'm going to lower that, but our Km is going to stay the same. And so our Km originally was 1.5, so let's add this onto the plot. I don't remember what color. Okay, green. Now our Vmax it's definitely going to hit our Vmax, so there's our green Vmax, it's here. And our Km is the same. So we see half our Vmax there actually matches up. So it's hard to see, it's covering them both up. So there's a blue Km here, so our Km stay the same, okay, but our Vmax drops. Now uncompetitive is actually kind of like both of these things. And so we would end up with a lower Vmax and a higher Km. So it doesn't grab it as well, and it doesn't work as well in general. And there's in purple, and so you can see these keep on dropping. And so that's just one way to visualize it. I encourage you to go here and play with this website because it's pretty kind of fun. To add just another way to visualize this, I want to jump back to the video with the robots because she's going to now talk about the at least the competitive and non-competitive types of inhibitors.
When a competitive inhibitor is present, such as the stainless steel fork, our robros can't distinguish between the inhibitor and their substrate. They treat the stainless steel fork like a plastic fork, but can't convert it to a spork. This slows down the overall system, so the apparent Km of the system increases. A non-competitive inhibitor, like this chip, decreases the robot's efficiency at converting forks into sporks, but has no effect on their ability to bind forks. So the overall Vmax of the system decreases, but the Km remains the same.
Hopefully that helps. I always like watching that. It makes it make sense in my brain. If you're into calculus, which I am not, you can get real deep into the equations behind enzyme kinetics. As far as asking questions about that, that's not really in my wheelhouse. I don't like doing that. I don't really see how it's super helpful. But there's different ways to approach it. And the only reason I put this on here is you'll see this visualization often where you have your enzyme in E and then your substrate in S. And then here's our ES complex, okay, stuck together. And then there's our enzyme plus our product, okay? And then in I will be the inhibitor. And so in this case, this would be a competitive one. So it's binding to the enzyme's active site. You can have a non-competitive one, and then you can start to combine things. And so that's where the mixed equations come from. And that's where I just liked this because it was nice. It's visual for me, for dummies like me that need visuals, not that we're dummies. And then for calculus nerds, they can look at this. But they're massive, giant equations, and the equations actually get even more complicated when you move on to allosteric inhibition. And so this would be, so when you have non-competitive inhibition, so it's binding somewhere else on the enzyme, that's what this would be. Okay, so when you have non-competitive inhibition, if it's allosteric though, and so this is, if you took micro with me, this is what we would talk about with like allosteric inhibition or allosteric activation. They get complicated, so a lot of times they're glossed over in biochemistry courses, as I'm doing right now. I'm kind of glossing over it, but allosteric enzymes, basically, they have one or more ways to either turn them on, i.e. activate them so they do their job better, or to turn it off. And usually what it's doing is it's just changing the active site shape. And so if you have an allosteric inhibitor, your enzyme is working fine, and then you throw the inhibitor somewhere, and then it changes the shape of the active site, and it doesn't do its job as well. Okay, so that would be this. I'm not really changing the Vmax though, which I realize is different for non-competitive. Oh man, I dug real deep and got real lost is the wrong word, but just I did not think it was relevant to talk about the differences between allosteric and non-competitive inhibition. If you would like to dig deeper, I can help you with that. However, for this class, you don't need to. Allosteric activation, same idea, it's just the opposite. So when I take my activator or whatever and I stick it in there, it's going to make the active site shape properly. So that's an allosteric inhibition, allosteric activation. Not the same as non-competitive inhibition, but similar concepts. The actual kinetics are interesting. and get real complicated real fast, which scares a lot of people away from them. So now, I wanted to talk about something very specific. Okay, I want to get back to some of the more practical aspects of what we're talking about. I want to look at an actual enzyme's active site and show you what's going on. Get into this. So I wanted to talk about chymotrypsin. So chymotrypsin is an enzyme. In the blue here is going to be our enzyme, and then here is our active site. So it's got some really cool structures to it. And I wanted to talk about this a little bit. So what it's going to do is it cleaves proteins, okay? It cleaves polypeptides, and it likes to cleave them when it has a nice big hydrophobic residue, okay? So in this case, it's phenylalanine, okay? And phenylalanine has this nice big little hexagonal molecule here. It's gonna stick in that hydrophobic pocket right here, okay? And then the rest of the molecule is gonna kind of stick out this way. Okay. And then you can see we've got all these other, in our active site, remember the residues of the amino acids that comprise the active site that stick into the active site are really, really important. And they're going to be what actually drives the reaction forward, okay? Now I have all the text here, but I'm gonna kind of take you through what's going on. So our substrate is going to bind, okay? So this could be anything. So basically the rest of the amino acid can extend out this way, and it can extend out this way. And we're going to bind in here, and what we're going to do is we're going to watch as we break this bond right here. So let's keep on going. So there's our amino acid now, our polypeptide chain. Our protein is now stuck in here. So this would be no longer our enzyme separately from our substrate. It's our enzyme substrate complex. And what we're going to see is that we need to basically, we've got this carbon here with these 4 bonds, right? Carbon likes to have 4 bonds. And what we're going to do is we're going to take some of these electrons here, and we're going to move on to this oxygen, which sort of frees up the space of one of these bonds here for carbon to then grab onto this oxygen here. And so this oxygen, it's got its hydrogen here, and we're going to sort of pull some of the power, the electronegativity away from this oxygen, from this hydrogen, using this hydrogen bond here, okay? And so we're gonna pull some of that away, and that allows us to move these electrons onto this carbon, okay? And we're gonna form what we call a tetrahedral intermediate, okay? And so let's see what happens. So we're gonna watch these electrons are gonna jump over here. So now my oxygen has electrons here, okay? And then I've formed a bond here, right? So I didn't have a bond here, so we're gonna form it here. Right, so now I have 4 bonds to carbon. I've lost that double bond. And what I'm going to basically do is I want to take the bond here between this carbon and this amine group here. Okay, so this is a peptide bond, right? So we had our carboxylic acid on one side and our amine group on the other side, and we want to form a new bond to this nitrogen. And so I need to balance out those bonds. The point of what I'm showing you here is not necessarily to memorize all these steps or anything. It's to show you that these R groups, or the residues, these R groups are going to serve to functionally perform the actual action of the enzyme. Usually we're kind of zoomed out. We say, okay, what is this enzyme doing? It's cleaving this thing, awesome. And we're studying the reaction rates. But what's physically happening? Well, it's all happening super fast here, right? And so I can't balance out, I can't get this double bond to kind of flip out here and put some electrons on this oxygen if I don't balance out that extra negative charge of those electrons without these hydrogens here, which I can't do that without this serine and this glycine, okay, which are at their respective locations. It doesn't really matter. It's just that I have these hydrogens here that can form a hydrogen bond and sort of balance out that negative charge, okay? And so that's what we see here. So we've got these hydrogen bonds here, balancing out that negative charge. So we're kind of we're pulling that double bonded oxygen charge away so that we can allow the carbon to form over here and get that bond formed. And then we're going to separate and flip this bond over here. So remember, this bond's attached to this nitrogen. So basically, this bond is going to flip over here. The carbon needs 4 bonds, so this double bond is going to reform. Okay? And then this nitrogen, well, it's going to lose a bond as well, so we're going to form it onto this hydrogen right here. So let's see. All right, and so that's what we see, sort of. Oh, sorry, the product goes on here. So there's the NH2. So the nitrogen here grabs onto this hydrogen. Okay, there it is. So now it's an NH2. It was NH, now it's NH2, right? It's happy, we leave, and so this entire chunk of protein just got cleaved and it's gonna fall out, okay? But the rest of it is still stuck in this enzyme. and it's still stuck in the active site. And so what we need to do is kind of cap this little end right here and make it not look as weird. Because right now it's stuck. So right now, this end of our peptide, of our amino acid, is literally bound to the enzyme. So we call it the acyl enzyme intermediate because we're trapped, we're stuck here. So I need to now sort of restore the enzyme. And that's one of the most important things that enzymes do. Enzymes are able to be restored. That's really, really important. We need them to be able to do their job over and over and over again. So how we actually restore them needs to be very efficient and really easy to do. And so in this case, we're actually going to use water. So we're going to follow our water molecule. So there's our water molecule. It's going to come in here. We're going to put a hydrogen back here. Okay, so we have our nitrogen forming a nice little, what is that, a hydrogen bond here. And we're going to stick our hydroxyl group because we're going to have that hydrogen here, and we're going to take these extra electrons and stick them on this carbon. And hey, look at that. We're going to have to flip these electrons from this double bond over onto this oxygen. But the good thing, we've got this glycine serine here to kind of hold on to that negative charge. Okay, so let's see what that looks like. So there, we've got this bond reformed here. Okay, that should look familiar. This is what it's supposed to look like on this end. And then we need to restore the serine here. So we need to get rid of this bond because right now, my peptide is still bound to the serine residue, and that's not good. We don't want that. Something really interesting, if you read deeper into these things, the apparent, so I put this here to remind me. So this whole process in the active site, this active site actually kind of squishes. It crushes a little bit, which then changes the apparent pKa. So histidine has a really important pKa. Its pKa is, it depends on where you read, but for free histidine is between 6 and 7. Okay, so it's actually relatively acidic, but it's kind of neutral-ish. So if it's 6 or 7, then it's able to donate its proton really well. If I squeeze it together, that raises the apparent pKa. And so if I have a really high pKa, like 12 is what it raises it to. If I have a pKa of 12, I do not like to donate my protons. In fact, I like to take on a proton, and that's what we're seeing here. So we're seeing that as this active site is actually going to sort of crush itself, which makes it easier to form a bond here and to sort of restore the histidine back to where it was. And so we end up with this bond forms here. Okay, so we're going to flip this over and bond here, this hydrogen and oxygen there. So we have a little alcohol group there. And then we have this little nitrogen bound via a short length hydrogen bond. There's different kinds of hydrogen bonds. This one was a short one, but that all happened because the actual active site was crushed a little bit, and it allowed that to happen, and then you lose the product. And so this thing actually falls out, which is very, very cool. And so one last couple things I wanted to talk about was that when this happens, there's some interesting things that we can study. So I mentioned pKa. So the actual pH of where this occurs is really, really important. So let's look at that. People have studied this extensively. So over here, we're going to try and bring this all back together. Okay. So over here we have velocity. Just look at this. Look at A here. So we have my velocity, right? So that's the rate of my reaction, and there is a peak rate. Okay, based on pH. So I have a 6 to 10 pH, and at pH of 8 is the peak. Okay, and then it starts to drop back down. Okay, so we want my pH to be around 8. And so we talked about that very early on, that enzymes have specific pHs, right, that they like to function at. So my velocity peaks at 8. So why does it drop down here? Well, that's a good question. Why can't I operate well at a pH of 6? Well, let's come over here. So Kcat is just the dissociation for your enzyme substrate complex going to enzyme plus a product. So it's sort of your KD basically. And so in this context, that's a good thing. So because we want our enzyme to grab onto the peptide, cut it in half, and then throw off the pieces. Okay, so as our pH becomes more and more hydroxyl, okay, the histidine that we've been talking about, so that's this guy right here, okay, that histidine will become protonated. Okay, so as it becomes protonated, okay, so it works really well over here. Okay, it deprotonates really well. So as we increase our pH, that means it's becoming more and more acidic environment, and so it's going to start to protonate that histidine. So as there's more and more hydrogens floating around here, I can accidentally put a hydrogen here, which I don't want to do that until I'm ready, okay? I only want to do that using water, okay? So if this nitrogen here is grabbing onto a hydrogen without water, okay? So it's just kind of sitting there. Well, then this process is broken, right? I can't do this anymore because I want to get my hydrogen for this nitrogen from water. I don't want it to get it just from like the solution, which that's what this means. So if I have a really low pH, that means there's just hydrogen just floating around. And because histidine has a pKa of like 6 or 7, then if it gets really low, then this histidine gets protonated before I want it to be, okay? And so that's why it doesn't work at a low pH. Now, the opposite is a little different, okay? So in this case, if I raise my pH, so that means it becomes really basic, right? And so as it becomes basic, it starts to not work either, okay? So it's 1/km, but really it's just how fast is this reaction? How well is this reaction occurring? And what's happening here is a different problem, okay? So if we actually go back and we look at our N-terminus, okay? So let's back up here. So here is our N-terminus, or our amine group terminus here. And basically what happens is if it's too basic, this N-terminus, so most of the amino termini for peptides is on the order of 10 to 12, okay, for their pKa's. And so what they will do is, as long as they are under that, then they will, as long as they are more basic than the environment, so like 8, then they don't deprotonate, right? They will just accept protons, awesome. However, if the environment around here gets really basic, so imagine there's like tons of hydroxide ions just floating around, then this amine group might accidentally donate one of its protons, like this proton here, or when it gets protonated here, to the solution. Okay, so let's say it donates this hydrogen here to the solution. Well, now I'm kind of stuck because I've messed up the actual charges that are associated with this region, okay? So I need my charges to be very consistent, okay? I need a hydrogen here. I need this oxygen to have a nice negative charge here. And so if I start throwing hydrogens and oxygens in there, it messes with what we call salt bridges, but that's just charges, okay? So when you have hydrogen bonds and charges associated with proteins, we call those salt bridges. And so you start messing up that. And so as it becomes more and more basic, you lose that hydrogen, and thus you lose the ability of this enzyme to actually do its activity. And so that's why we have a peak enzyme activity here at pH of 8, because you're either protonating your histidine as you go this way, or you're deprotonating the amino group on the cleaved peptide. Okay, super interesting stuff. Thank you for putting up with me as we kind of go through that, but it is important. And I like this example a lot because it's kind of cool. Now, I want to show you one more video. It's from the protein database. It's the rest of that video, and it shows just one more example, and I really like it because it visualizes the active site of an enzyme. So let's take a look.
To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron-sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row.
Good stuff. I love it. Last thing I want to mention, we're going to talk about this later on, especially when we talk about glycolysis and the citric acid cycle. We're going to talk about this feedback mechanism. So when you have an enzyme doing its job, we have a whole series of enzymes. So think glycolysis, there's like 12 different steps. And so at each step, I'm making an intermediate substrate, and then a different enzyme does something and make another intermediate substrate, and then I make a product. And what can happen is you can have feedback, either inhibition or activation. And so you have different regulators. So the presence of an end product can then go back and affect the enzyme that's initiating the first step. A lot of times we think about this in terms of inhibition. And so, for instance, in glycolysis, the whole point of glycolysis is, well, there's 2 points: to make substrate level phosphorylation, so you make ATP, and to make some of the byproducts that will then go into the citric acid cycle and into the electron transport chain. Awesome. Well, if you're making a bunch of ATP, you don't want glycolysis to be doing its thing, because I have way more ATP than I need. It doesn't have a super long half-life, so it takes energy to make ATP, so stop making it. So if you have a bunch of ATP, it's going to actually inhibit phosphofructokinase, which is one of the enzymes in glycolysis. However, if I have excess AMP, okay, so ATP is 3 phosphates, AMP only has 1. If I have a bunch of AMP floating around, well, that means I've probably used up a lot of ATP and I'm ready for more. I need more ATP. And so AMP is actually an activator of phosphofructokinase. And we'll talk more about this when we actually get into glycolysis. Thank you guys so much for listening. I know this is a little bit longer, but we got through this whole topic. It would have taken forever. So thanks for being patient.